How do you insert a new gene into a plant? There are many different ways, and technology is always changing. But let's explore the two most common ways, through the gene gun and through agrobacteria-mediated transformation. No matter the delivery method, the first step is to engineer your gene into a small DNA construct that can be delivered to the plant. This construct is often referred to as the vector. In this vector, you need to include your gene of interest and all of the regulatory elements that you want to control the expression of this gene. A promoter sequence signals when, where, and how much of your gene should be expressed in the plant. The construct may also contain a selectable marker, such as a gene for antibiotic resistance, so that successful transformation events can easily be detected. Once you have created your DNA delivery vector, you need to move it into the plant using a plant transformation method. When it works, agrobacterium transformation is usually preferred by scientists. In this method, scientists take advantage of a naturally occurring transformation system. In nature, Agrobacterium tumefaciens, a common pathogenic soil bacterium, transfers some of the genes it contains on a plasmid to wounded plants. It inserts a part of its DNA that causes the plants to produce amino acids called opines that the Agrobacterium pathogen can use to sustain itself. In the lab, scientists remove the genes for opine production and use molecular scissors called restriction enzymes to insert into the vector the genes that they're interested in studying or have the plant express. Agrobacterium can be caused to carry that vector and deliver the new gene of interest into the plant in the same way it does in nature, by moving a fragment of its DNA containing the gene of interest through the plant cell and into the nucleus where it integrates into the plant genome. The plant now carries the new gene and will read its code just as it reads its own native code. Cells that have been effectively transformed with the new genetic material are then selected. Full plants are regenerated through tissue culture, and whole adult plants ultimately set seed to a second generation, some of which carry the new gene. A second method of transforming plants is through the gene gun, also called biolistics or particle bombardment. The gene gun is often used to transform the genomes of organelles, such as chloroplasts. The gene gun is also used to transform plants that are recalcitrant to transformation by agrobacterium, such as wheat. In this method, particles of gold or tungsten are coated with the DNA of interest. These particles are then shot into cultured plant cells or plant embryos, physically forcing a small percentage of the DNA through the cell's membranes and into the compartment containing the target genome. At some low frequency, the gene of interest will incorporate into the target genome. Successful transformations are detected using a selectable marker, such as antibiotic resistance, on tissue culture media. Transformed cells are then grown up into small plants through tissue culture. Eventually, those plants can be transferred into the soil and can continue to grow and set seed. So let's summarize. To make a transgenic GMO plant, you must first create your DNA construct. Then, you choose your delivery method, agrobacterium-mediated transformation or the gene gun. Next, you deliver to the appropriate plant tissue. Cells, embryos, or leaf pieces are examples. Then you regenerate your plants in tissue culture. Once the plants have generated sufficient roots and shoots, they are ready to be transferred into soil. Eventually, the plants set seed, and a new generation of plants carrying the trait can be harvested.